just so you know.
Yeah, right?
All right. So a couple of things I want to cover before we get started on today. We'll get through what we can. Wherever we stop, that's what the last part of the exam is. First thing I wanted to cover are what are the objectives, and then to let you know to bring a calculator with you on Monday. You know, bring a calculator. There's one problem that you actually have to do math for. So in terms of the objectives here, everyone from control of growth, microbial growth, and then antibiotics is presented some way on the exam except for one of them, so I'll show that in a second. But the first one here, 5.1, identify the 4 stages. This is basically, can you identify lag phase, log phase, stationary, and death phase? Can you describe them, or could you pick out the definition of one of them if one was provided to you? That's that first objective. The second, classifying. This is what are the terms that we use to describe the organism over a range of temperatures, salt concentrations, pH values, and then finally, based on their oxygen or their growth in a thioglykylate tube. And then 5.3, how does growth affect it? This really correlated then the thioglykylate tube to also the metabolism of the organism. So can they perform fermentation? Could they perform anaerobic respiration? Could they perform aerobic respiration? And then also ties it together with the enzyme catalase. And just a reminder of what catalase is. Yeah, it breaks down hydrogen peroxide, but it's one of a number of proteins, along with superoxide dismutase that we didn't focus as much on, that are involved in breaking down reactive oxygen species that get created from just the metabolism of the microorganism. So if an organism lives in the presence of oxygen, it has to have catalase. The basic premise here. Know the basic structure of the spore. There's really not much that I want you to know about this other than it's got a thick coat, it's protected against environmental stress, these are highly resistant. What kind of organisms produce spores? Gram-positive bacillus are the only ones that produce spores. It's not even all of them. And then the last two, describe methods to measure growth. Basically, have a basic understanding of, like, when we do spectrophotometry, we use light to measure how much bacteria there is, and then if you can at least remind yourself how plate counting and serial dilutions work, that'll definitely help you on the take-home one. Okay? And then calculating and evaluating. Basically, this kind of goes back to the paper that we read. Can you look at a graph and determine was growth inhibited or not? If so, by how much? And also, does that mean that something you're using to promote growth or inhibit growth is effective? The take-home question is basically that last bullet point. Okay? So the take-home question will be on your evaluating, do the filters that we tested in lab, did they work like they claimed to? Okay. I'll be posting that a little bit later today, and you'll have until Tuesday night to finish it. All right, so next one, control of growth. So describe physical and chemical controls. I guess I came to the realization last night that the chemical controls, there's really not that much I want you to know about them, because every year I'm deciding more and more that it's less and less important to cover every single one of them. What I do want you to know is, in the context of a problem, whether something would be considered an antiseptic, a degerming agent, a disinfectant, or a sterilant. That's what I want you to be able to determine. So, most important thing is remember or start to try to memorize or recall what is the difference between a disinfectant and an antiseptic. What makes both of those different from something that is a sterilant. In terms of the physical controls, we've talked about a number of those. We've talked about heat in the form of sterilization with an autoclave or pasteurization. We've talked about filters. We've talked about radiation, both in the form of UV and ionizing, and they're damaging DNA. We've talked less so about acids. We've talked a lot about affecting temperature, a little bit about desiccation as a way to remove water or to raise salt concentrations. And in essence, what's the storyline here? If you're using a physical measure to inhibit the growth of a microbe, how do we do it? Do anything that isn't optimal for the organism. Okay, that's the bottom line. And that really gets down to 6.3, developing these strategies for exploiting microbes' metabolism. So if it's an acidotolerant organism, obviously adding acid or encouraging some sort of lactic acid fermentation to inhibit the growth of the organism, it's not going to work. It's tolerant to that condition. So in part, this should be intuitive. And then the last one here, again, 6.4 actually goes a lot along with 5.6, using bacteria data to evaluate whether an antimicrobial is effective or not. I should also include that the other ways we can do this are by performing minimal inhibitory concentration or broth dilution experiments, and also by performing those disk diffusion assays. So, and we've seen that again with antibiotics, that we've been doing those measurements as well. So those same type of tests can apply for chemical. And then for the last set here, I haven't crossed out the last one yet, but that one we will come back to. So we're going to talk about antibiotic resistance today, but we're going to talk about it in the context of what's happening at the cellular level. That's a concept we'll actually come back to in probably about a week and a half. And then probably in 3 weeks from now, we'll talk more about the social practices that lead to antibiotic resistance as well. So that was supposed to be today, but we'll push it off. We'll talk about 7.3 today, and then the other two, justifying the use of an antibiotic given its spectrum, molecular target, therapeutic index, effectiveness. Basically, the things that I've said for this are memorize that chart. So make sure that you know the targets for the different antibiotics and that you know the pathways that are inhibited. And then we talked about how, for example, we want to choose drugs that, if we had a choice, would have a higher therapeutic index. And that is dependent on the toxic dose and the therapeutic dose. We'll come back to the importance of spectrum, I think, later. That becomes an important topic when we start to talk about some of these social practices as well. So that'll be something for later. And then last but not least, analyzing results for effectiveness. Again, Kirby-Bauer assays, disk diffusion assays, E-tests, and then also on top of it, determining minimal inhibitory concentrations to determine how effective they are. So, yes, we've covered a lot, but I don't know if you've noticed it or not, but a lot of these things overlap. A lot of it overlaps. In our conversations of chemical growth, we've come back and we've revisited some of the same tests. These topics will come up again down the road. We will continue to work with these. This isn't the last time you'll see them, whether you like it or not. So I'll pause real quick. Any questions about what's on the exam? Abby? Yeah, I want you basically to know the stuff that relates to the antibiotics. So gyrase is involved in replication. If you inhibit gyrase, you block replication. If rifampicin blocks RNA polymerase, it blocks transcription. And then the last one, for the nucleosides and chloramphenicol, if they block the ribosome, they block translation. Part of doing that ahead of time was actually to get you thinking about stuff that we will start on Wednesday. I also needed to fill in a day since I wasn't going to be here, and I thought that was a good on your own. So the gyrase and its alternate replication. Its role in replication. So if you do the gyrase, replication stops. So the quinolones regulate that. Rifampins or rifamycin or any of those of that class target RNA polymerase. And then the last one would be the ribosome inhibitors, glycosides and chloramphenicol. So the format for this one is somewhat similar, somewhat different than the last one. What I have done is I've actually just created one glorified case study on one single bacteria. So every question pertains to this one bacteria, and that means that your previous answers might help you with answers further down throughout the whole exam. Okay, so make sure that you keep that in mind as you're looking at it. If I'm not mistaken, I'm trying to run through the numbers in my head real quick, but I think that there's 6 multiple choice on this one. There's somewhere on the order of another. I think somewhere between 10 to 20 like fill in the blank or like single word answer type problems. Those and the multiple choice, because this test is worth more, each of those is 2.5 points apiece. And then there are 4 or 5 shorter response that should be able to be completed within 2, 3, 4 sentences for most of them. I'll be going through it today to see the length of it, so it might be slightly modified to make up for it being too long. 85 points in class, the remaining 15 will be the take home. Any questions? Yeah. Yep, that is the plan. I will be here a little bit early on Monday morning, so when you guys come in, you can begin to take it right away. And we'll do the same thing as last time. We'll do space out every other row so that I can make sure I can get in between folks and then answer questions. All right, let's go ahead and discuss. I want to actually pick up with one of the things that we left off with on Wednesday. Oops. So when we left off on Wednesday, we were talking about the minimal inhibitory concentration. And we said it's the minimal concentration of the drug that is needed to completely inhibit the growth of the microbe. And typically, we do a broth dilution series like this to analyze that. So in this case here, we said antibiotic X has an inhibitory concentration of what? 4, and then for this bottom one, we said it would be 2 if these were 2-fold dilutions. So the next question, one of the follow-up questions is, how do you identify what we call the MBC, the minimal bactericidal concentration? So bactericidal, well, if something is cidal, what does that mean? Kills, death. In other words, this is the minimal bactericidal concentration is the minimal concentration of the antibiotic to not only inhibit growth, but to kill all of the microbes. So for something like this, you have to do a modification of the experiment. One way would be to actually do serial dilutions and plate counts to determine, well, in which of these do we no longer see any more bacteria? There's an alternative way that you can do it as well, and it's a method of subculture. I'm going to draw this one up on the board today. So from each of these wells, or at least those that have no bacterial growth, let's take antibiotic Y, for example. So growth, 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 these last two. To determine the minimal bactericidal concentration, we have to take bacteria out of these and determine, are they going to be able to grow? Because if they don't grow, they're obviously dead. Okay? But we want to do that without selection. And so in other words, to determine the minimal bactericidal concentration, we actually have to grow these cells again out of these cultures, but in the absence of an antibiotic. So we would take basically 5 new wells and add into each of them just media, LB. And ask, in 24 hours, do we observe growth? If we do, the cells were originally inhibited, but the concentration of the antibiotic isn't high enough to actually result in the death of those cells to be considered bactericidal. So, for example, what if we saw results like this? So after subculturing, taking a little bit of bacteria from each of these, adding it with some fresh LB, we find the results on the bottom there, growth in just the last well. What's the minimal bactericidal concentration? 4. It's the only one in which we don't observe growth. And again, this growth would be occurring in the absence of any sort of selection. So we've gone from a culture that has antibiotic, we've taken our inoculating loop essentially, and moved it in, inoculated a new fresh culture with just nutrient-rich media. Mueller-Hinton as well would be used. We observe growth in all of them except for the last one. The reason why we don't see any growth is because the cells died up here. These ones, though, those in the presence of 2 milligrams per mil of the antibiotic, they were just inhibited. They were still alive, but the concentration was high enough to inhibit them from observably growing. And so when we remove them from the antibiotic and culture them in fresh broth, they're able to start growing again. I think one of the things that this kind of hits home again is that one of the points we made really early on is that lack of growth or the inability of a microbe to grow doesn't mean death. And this kind of reiterates that point. We can have concentrations of salt, for example, that are high enough to inhibit growth but aren't bactericidal to the organism. Questions on that concept of identifying these or performing this experiment? Okay. Given the toxic dose that is here for antibiotic X and antibiotic Y, given the therapeutic dose, which we'll just assume in this case is the same as the minimal inhibitory concentration, which is the better one to use? So which is the best antibiotic given the therapeutic dose and the toxic dose? I'll give you a minute to look at this one to figure it out. How do we calculate therapeutic index? What's the equation we use to calculate it? Toxic dose over therapeutic dose. So for antibiotic X, it's 2/4. Is that right? Just a half. For Y, it's 0.25/2. All right on that? Yeah. Okay. Which is 0.125. Given that data, if you were to prescribe one of these antibiotics, which one would you prescribe? X, larger therapeutic index. In other words, the concentration at which it's being taken relative to the toxic dose, that ratio is better than what we observed for antibiotic Y. In other words, what that means is that the likelihood that it's going to cause toxic side effects to the individual who's taking it are going to be less than they are for antibiotic Y. So that's how we would use that therapeutic index for some of these. Now, there are always other considerations that we need to make. So, for example, this therapeutic index can change depending on whether it's a child versus a young adult versus someone who is elderly. It depends on it can depend on the individual's metabolism of the antibiotic as well, which can't always be calculated on a per person basis. So these numbers are largely based on large populations in which the study of these antibiotics have been performed. All right, with the time that we have left, I want to cover antibiotic resistance. I want to cover this from mechanisms that microorganisms use that can lead to resistance. So we will talk later on about how resistance can be developed through mutations. That is a conversation for a later time, but that's actually one of the mechanisms by which bacteria can gain resistance to an antibiotic. It's just through either spontaneous or random mutations, or ones that occur as a result of, for example, exposure to UV light. We're going to talk about two major ones, but let me just cover the four examples real quick. The first two of them actually refer to like a modification or an inactivation of the antibiotic. So one is hydrolysis. Remember, for hydrolysis reactions involve what? Water. Water breaks a bond. So by a hydrolysis reaction driven by an enzyme, that can lead to the inactivation of some antibiotics. In fact, this is one of the most common ways in which the beta-lactams are inactivated. And we'll talk about that specific example. Another way is through modification. Some organisms, some microorganisms have genes that encode for an enzyme that adds an acetyl group onto chloramphenicol. Basically, that modification inactivates chloramphenicol, prevents it from binding to the ribosome. So both of these involve some sort of modification. One's an addition of some molecule onto the antibiotic. The other is a cleavage or breaking down of the antibiotic. I'm going to skip a slide real quick, and we'll come back to the beta-lactams. The last two mechanisms are target modification, aka mutation. We'll cover this in a week. The last one is efflux pumps. So you remember, particularly gram-negative bacteria have outer membrane proteins, or porins. And some of these porins are actually active transport proteins that take, as soon as antibiotics come into the cell, these are part of a multi-protein complex that takes those antibiotics and using ATP, pumps them right back out. In other words, the antibiotic never comes into contact with its potential target gene. And one of the things that we will talk about over the next few weeks is a multi-drug resistance pump in Pseudomonas aeruginosa. one that has been found to be highly important to Pseudomonas' resistance to a number of different antibiotics. Let me see if I can pull something up real quick, and I'll give you an example of this. There it is. Okay, so this is, oh, that's horrible. Basically, what this is it's a paper that was, or an experiment that was performed in which the researchers took a prototypical strain of Pseudomonas aeruginosa. It's called PA01. The most important thing to know about it is that most studies that are done in laboratory settings use this particular strain of Pseudomonas as kind of a model for all Pseudomonas aeruginosa strains. Pseudomonas is worrisome for a number of reasons. We'll talk about it in the context of cystic fibrosis later on. And in this particular paper, what these researchers were doing was looking at how Pseudomonas aeruginosa responded to different antibiotics, or in fact determined what the MIC for each of these antibiotics were in wild type cells, so just the normal PA01 strain, or in cells in which they had knocked out, we're going to look at this one right here, this gene known as OPRM, and this is that outer membrane pump protein. What I want you to do real quick, don't look at all these, there's a lot of data here. Focus on trends, again. But take a look at, for example, norfloxacin, ciprofloxacin. Those are both quinolones. And take a look at tetracycline, chloramphenicol, which both target the ribosome. But I want you to compare the mix between the original strain and the strain that doesn't have OPR. Can everybody see that okay? Do I need to zoom in some more? What's the general observation? So in the absence of OPR, are the mix higher or lower? They're lower, which means what? These cells have become more sensitive or resistant to these antibiotics. They're more sensitive. It takes less of the antibiotic to inhibit their growth. Therefore, they're more sensitive. And in fact, what has happened here is that in the presence of this when this particular gene has been deleted, here's a normal cell, here's that foreign pump, pumping those antibiotics out. When this is gone, it basically locks the antibiotic inside of those cells. So now there's no way for Pseudomonas to pump these antibiotics back out. This is a really important point, because one of the things that we'll talk about in a couple of weeks, I cannot wait to talk about this paper. I saw somebody talk about it this summer. Basically, what they have done is they've used phages, bacterial viruses, as a way to mutate that particular protein. And what they show is when they mutate that protein, the cells are now more sensitive to antibiotics. They've used it in a hospital setting. I mean, this is like earth-shattering, because we're talking about how frequently resistance to antibiotics is becoming right now. This is really important stuff that they're doing. So we'll come back and talk about this, but I wanted to just give you an idea of one of these ways in which resistance can occur, is through these pumps. Okay. Questions on that mechanism? It is higher in the wild type. Yeah, so the wild type are the original cells. These ones here are the original cells minus the OPRM gene or that pump protein. So what we're, yeah, so they don't have it. So what we're saying is the wild type, this is normally what we would find. It's quite resistant to novobiocin and chloramphenicol and tetracycline. When you knock out that gene, the mix decrease, therefore the cells are more sensitive to these antibiotics. That's what we're saying. Let's take one more example, and this is a classical example. I'm going to show you two molecules real quick. Amoxicillin on the right, clavulanic acid on the left. Both of these are what kind of antibiotic class? Got this lovely ring structure right here. Classic for beta-lactams. In other words, what do these target? What is the target of the beta-lactams? Penicillin-binding proteins, and they block what? Cell wall synthesis. Amoxicillin is a true beta-lactam. Clavulanic acid is actually what we call a beta-lactamase inhibitor. And this is the last class of antibiotics I want you to know. So I'm throwing one more at you. The beta-lactamase inhibitors, as the name implies, inhibit a protein known as beta-lactamase. Anybody want to guess what the substrate for beta-lactamase is? It's in the name. What is a substrate for a beta-lactamase? A beta-lactam. And we talked about, or we already looked at an example. Here we go. We said one of the ways that some of these antibiotics work is by hydrolysis. This is what the beta-lactamase does. It takes a beta-lactam antibiotic and it hydrolyzes that beta-lactam ring structure. And in doing so, beta-lactam antibiotics, penicillin, etc., no longer function. So here's the exact reaction. I'm not expecting you to know this. Basically, water is added to this carbon-nitrogen bond right here, creates what we call penicilloic acid. Basically, this is inactive. This no longer looks enough like the substrate to add so that it can fit into the active site and create an irreversible bond with the penicillin binding proteins. The function of the beta-lactamase inhibitors, as the name would imply, is to inhibit the beta-lactamase. In other words, you are inhibiting an inhibitor. Okay? So, what we find is that organisms in response to beta-lactams can gain resistance in a couple of different ways. I'm just going to draw the ring structure here. We can find resistance to these beta-lactams in a couple of ways. One is by mutation or change in the amino acid sequence of the penicillin binding proteins that now makes it so that penicillin can't bind to the active site, but the enzyme can still perform its function. That's one mechanism. Another is through these efflux pumps that Pseudomonas encodes. The third is through inheriting a gene known as the beta-lactamase, or the BLA gene, BLA. This gene, as we just said, its job is to take this from an active form to an inactive one. Well, in the presence of clavulanic acid actually, we took a look at this already. It looks a lot like these beta-lactams. In fact, it's got the same classical ring structure of a beta-lactam. So I'm going to abbreviate this CA. Clavulanic acid fits into the active site of the beta-lactamase and inhibits the beta-lactamase from targeting the active beta-lactam. In other words, we've got two competitive methodologies here. The competitive inhibition of the beta-lactamase now allowing for these penicillin or beta-lactams to now go ahead and inhibit the function of those penicillin binding proteins, or those PBPs.
Okay, can you repeat that? So the CA fits into the active site of beta-lactamase, and then what?
And in doing so, now the beta-lactamase can no longer, if it's binding to clavulanic acid, now these beta-lactams, they're not going to bind to the active site of the beta-lactamase because it's already occupied by clavulanic acid. They now can function as they were intended to be, as inhibitors of penicillin binding proteins. In other words, we're preventing, with these beta-lactamase inhibitors, we're preventing these cells from inactivating the beta-lactam antibiotics, penicillin, for example.
The CA is for the clavulanic acid is the one that like stops the bacteria.
Exactly.
The antibiotic.
Yes. Yes. So it's the one that inhibits the beta-lactamase from cleaving or breaking down the beta-lactam antibiotics. Let's take a look at how this would actually appear in an experiment. Let's think about this in two ways. Let's start with the Kirby-Bauer assay. So here's my plate. Let's imagine it's covered with bacteria already. On one disc, I add penicillin. On another, I add penicillin, and we'll add clavulanic acid. So we'll call it PCA. Let's imagine that our zone of inhibition around penicillin is something like this. Very, very small. If this organism has beta-lactamase that allows it to be resistant to penicillin, how would the zone of inhibition look when we add clavulanic acid and penicillin at the same time? Would it be similar in size, less in size, or greater in size than this particular zone of inhibition? Greater in size. Why, Derek? Good. Exactly. So that I would consider to be a good response. If we're looking for a complete one here, it allows it to inhibit growth because the clavulanic acid binds to the beta-lactamase, inhibits it from binding to penicillin. Penicillin can now bind to the penicillin binding proteins and inhibit cell wall synthesis. Good. Think of another result here real quick. What if the result looked like this? What if it were identical? Likely, yeah. It's likely another form of resistance. Why would you say that, Abby?
Because it doesn't work. Like if the clavulanic acid is not isn't working, it means that the beta-lactamase is probably not the issue.
Perfect. What could be these other forms of resistance? What's a likely explanation? Possibly the organism has an efflux pump that pumps out penicillin. Alternative would be mutations in the penicillin binding proteins themselves, so that they can still perform their cell wall synthesis but not bind to penicillin. What I want everybody to do now is draw out a mock result for the following. set up a small-scale broth dilution experiment. Let's say we've got 16, 8, 4, 2, and 1 microgram per mil of an antibiotic. On the top we have, let's say this time it's amoxicillin, which is another beta-lactam. And on the bottom it is known as augmentin. And augmentin is a combination of amoxicillin plus the clavulanic acid. What I want you to do is first, let's say that the minimal inhibitory concentration for amoxicillin itself is, let's say that it is 8 micrograms per mil. Okay? So what I want you to do first is then draw what the results are going to look like for amoxicillin. And then assuming this organism has a beta-lactam, or a beta-lactamase enzyme, draw what the results would look like for augmentin, or what you think they might look like. I can say that again. So we're going to say that for amoxicillin, the MIC is equal to 8 micrograms per mil. So part 1 is draw the results. And then part 2 is assume this organism also has a beta-lactamase, draw the results for augmentin given that it has a beta-lactamase. Take a couple of minutes. So it's, in other words, it's inhibiting.
Okay, so then what's that? CA mods penicillin binds to beta-lactam. Okay, so they work together now. Okay,
Yeah, so there's a synergistic effect in some ways, really. Okay, so in, oh goodness gracious. In the case of amoxicillin, then, if our minimal inhibitory concentration is 8 micrograms per mil, in which wells will we observe growth? 16. Any others? Nope. That's the only one. Good. I think we're good there. What might the results look like for augmentin if this organism had a beta-lactamase? And somebody can just tell me, like, where did you draw, what was your MIC that you had? So if this organism has a beta-lactamase, amoxicillin is a beta-lactam. Beta-lactamase does what to the amoxicillin? The MIC would change. The MIC would change. So, Kaylin, you're saying you've drawn that there's. Okay. Or wait, no, I did this wrong. I did this wrong, guys. I do this every single semester. Son of a gun. Yeah. Sorry. My apologies. No growth here. There we go. My apologies. My apologies. Every damn semester I screwed that up once. Damn it, almost made it. 5 minutes. So we get growth in all but the
Why is there growth right?
There's not. Thank you for questioning. Always question authority. If the MIC is 8 micrograms per ml, we'd see no growth there. We'd see no growth with 16 either. Okay. God bless.
I think it's usually.
Yeah. My apologies. Thank you. That's exactly why it was. All right. So with augmentin, then, Kaylin, you're saying that we wouldn't see growth in any of the wells, possibly. So if this organism has a beta-lactamase, the beta-lactamase inhibitor, clavulanic acid, would inhibit the organism's ability to break down amoxicillin, now allowing amoxicillin to inhibit penicillin binding proteins. In other words, there's a synergistic effect here. The combined effect of these two drugs is greater than either of them alone. So if that happens, does the sensitivity of the organism go up or down? Does it become more sensitive to the combination of these drugs than it was with amoxicillin alone? And the answer is yes. If it becomes more sensitive, what happens to the MIC? It goes down. It goes down. So we might say like a mock result, and this didn't have to be the one that you had, could have been that. Could have been that, the MIC is now 2 micrograms per ml. Or you could have chosen 4 micrograms per ml. Both of those would have been correct answers. Again, given the fact that we're trying to model here, the organism is more sensitive to the combination of these two antibiotics. There is a, from this last lesson, so antibiotics lesson 2, there is a good, better, best question that is directly related to this. So if you want to review, that would be a good place to start for it, okay? We've got 3 minutes. So, any last minute questions? All right. Try to enjoy the weekend. Try to find some joy in microbiology. Meaning, drink a fermented beverage. Doesn't have to be alcoholic. It could be kombucha if you would prefer that. All right. I'll see you guys on Monday. Have a good weekend. Yeah. Yeah, I'll be around from 9 to 11, or now to 11, and then like 2:30 to 4. Sure. Sure.
With sterilization, have you seen pictures like, and I got
Because what comes out the other end is sterile, is lacking growth. But with that said, it's not like an autoclave where you're killing everything. You're basically just trapping the microorganisms in the filter.
okay. And then also radiation is one of them sterilization and one of them.
One of them can be sterilization. UV will only work on surfaces. Ionized connection. So usually, and most of what they do, UV just like creates the ion primers. UV actually creates double-stranded breaks. Both of them result in mutation, and that's small like
This?
I missed that.
Okay, I was wondering.
It does.
So like, this was
The main difference is cycrotropes usually optimally grow around 20, 22 room temperature. Cycrophiles refrigerate. So like cycrophiles will best grow at about 4 degrees Celsius. The range is like -10 to +10. Cycrotropes, they can grow in refrigerated temperatures. So some of them can grow all the way down to 4 degrees, but their optimal is right around 22 or so. Once it gets to body temperature, they don't grow anymore.
Oh, this, like, So do those catalase and superoxidase dismutase work together?
They do.
Okay.
So really, it's kind of a two separate step process where SOD works first to break down those free radicals,
Okay.
hydrogen peroxide forms, and then catalase breaks that down.
Okay. That's what I was thinking. Sure. And then
And to this last one, yeah. So for this one, the two would remain the same. You would actually just multiply. So it would become like, say you started with one cell. The full out equation is 1 times 2 to the n, where this number represents how many you start with.
Okay.
So if you started out with 100 cells, it would be 100 times 2 to the n, where n kind of equals the number of generations.
Okay.
Yeah.
And then this, I just had a question. So on your key, it says that this is a mesophyte.
Yeah, that is definitely.
Okay. Okay.
So yes, find that too.
Okay.
Hi, Megan. Yeah. So if the organism has a beta lactam, you have to process your.
Oh wait, but then it'd be more sensitive, right?
Yes.
Okay. Okay, so then I just had a really high percent.
Yes.
Okay.
Yeah.
I think I was
That's-
looking at it as that. And then
You were yeah, and the thing to remember is that as that zone of if the zone of inhibition is if you have two ZOIs, zone of inhibitions, a small one and a large one, the one that is large, the MIC is going to be smaller.
Okay. Thank you.
Yeah.